Okay, so I guess the 5 minutes have passed, so we can start the session. Hello, everyone. My name is BAIBA VILNE, and I will be your chair today in this session related to artificial intelligence and big data and precision medicine. I guess the three topics do not need too much introduction, as these have been the big three of the recent years in biomedicine, and indeed quite a broad topic, so obviously we will not be able to cover every aspect, but we will start and touch upon genomics, big data registries, moving to image analysis, and finally arrive at digital twins. But before we start with the first speaker, I would like to remind you that we have a special format for this session, meaning that if you have any questions, you will have to write them down or keep in mind, because afterwards, after all the talks, we will ask our speakers to kindly come to the podium and take these 4 chairs, and then we will start the question and answer session. So, no questions during the talks, so to say. And with this, please let me welcome our first speaker, which is Tero Hiekkalinna. He's an associate professor and senior researcher at the Finnish Institute for Health and Welfare in Finland. His research is focused on population genetics and the development of simulation and analysis tools for genetics epidemiology. So please welcome.
Thank you for the organizer for invitation to have available to speak of my recent project, which I have been working last 2 years. So basically, my team has been working on simulating 1 million genomes with the phenotypes. So the background for this project is that the 1 million genomes initiative, which brings together multiple EU countries, including UK and Norway, the goal is to collect total of 1 million genome sequences from Europe. But in order to handle such data, the federated databases and system have to be developed, and they have to have some kind of test data, real synthetic test data, in order to test their system and validity and how those can be analyzed in those systems. So this project was organized by THL, funded by the Ministry of Social Affairs and Health Finland, and of course now partly funded by the European Genomic Data Infrastructure, GDI. And of course, important partner over here in this project is the CSC, which is the IT Center of Science Finland, which is providing all the computational services. So the team is over here, me in Kainu over there, and then research professor Markus Perola, who is next speaker over here in this session, and then the statistical brains and mastermind over here is Joseph Sterelvileker from the Columbia University, also a long-time collaborator of ours. And then we have wonderful programmer as well in our team, which is the computer science student Tero Hiekkalinna. So, why there's need for simulated genome-phenome dataset? So basically, simulated data can be used for benchmarking of the service implementation, technical standards, compatibility, and best practices for sensitive human genome data. So the purpose of this project is to create really anonymous datasets which can be freely used in testing those systems. So there's no problem if there's some kind of problem in the system, because this is simulated synthetic data, but it's real as possible. So if there's a problem, there's no problem if the data is somehow lost. But anyway, because this is based on the real data, it has all the characteristics as real data. So why do we simulate? What's the purpose of simulating population genetic data? So in the simulation, we attempt to mimic the evolutionary process, which generates all the genetic architecture which we see in the populations. Mate choice, migrations, population substructure, interaction with the environment. And of course, when you are simulating genomes and phenomes, you know exact models, and then you can test study designs, statistical approaches. So can your statistical approaches find any signal from the simulated data when you know exact truth how the disease has been simulated in the dataset? And of course, when you know the truth and simulate those, then it helps us to design what kind of sample designs, case control studies, family studies, or joint studies, and then what's the sample size that you can detect something, and when you have hypothesis of some disease, and then you can test your hypothesis over there, and you can select what is the good analysis method of your choice. And of course, because this is simulated based on the real data, there's no data protection problem as well. And of course, simulation helps guide your, gets your valuable results and guide you in your analysis of the real data. So how do we actually then do the simulation? So it's based on the chromosome-based simulation method published by Tero et al. Already in '93. So it based on the rapid simulation of population of few hundred generations of drift, but we are keeping track of the recombination event in each meiosis. So basically, when we have a mate choice and everything over there, we only keep track of each recombination point in each individual in each chromosome. That's the information we keep, of course, including family information as well. But we are just tracking the recombination points in the genome. And this information then can be used for generating those, because in the top level there is a chromosomes, and then there is a recombination happens during the in each generation, and then in the end it's a mosaic of those founder chromosomes at the top level. So based on that information, we can create those mosaic genomes in the end. So basically, we are using the publicly available datasets in this simulation to have as a founder population. So here's one is simply illustration, how do we keep a track of the recombination. So there's a grandparents over there. They have a chromosome intact in this case, and there's a meiosis, and then recombination occurs at where the star is. So these kids, one chromosome, paternal chromosome, is a mosaic of grandparents' chromosome. So that happens in the next generation. So in the end, the last generation over there is a mosaic of chromosomes from the founder population. How do we then simulate that 1 million individuals? So we use the International Genome Sample resource, which is really available dataset, sequence dataset in the internet. And we use 5 different populations as a founder population in this study. But then in the founder population, we also shuffle chromosomes as well to make even one person more anonymous. And then we have generations, we start the simulation, and there's 15 generations of migration, random mating, etc. But then we split to 10 different populations, so that each subpopulation over there, the founder population, they have the same founder population for each subpopulation. And we stop the simulation for each subpopulation when the last generation has 1 million individuals. Now you can see that we have simulated already 10 million individuals in the last population. 10 population, each population have already 1 million individuals in the last generation. So now we actually sample from each population 100,000 individuals with the pedigree structure, and then we take those 100,000 individuals from each population, and then we have 1 million individuals total. So we have generated more mosaic genomes than we are sampling. So about some of the data sizes, so storage requirements. So basically 1 million genomes in VCF data format, it relies between 30 and 50 terabytes It depends on how much there is a variation and comparison and how you organize the data actually. So the computational challenge is huge. Already beginning of the simulation when we are simulating those mosaic chromosomes. So our really important collaborator over here, without we couldn't do this, is the CSC, which hosts the Puhti and Lumi third fastest supercomputer in the world. And I'm really excited because my background is in computer science. I'm a nerd, I'm a geek, so I'm really happy to use that supercomputer for this simulation study. And of course, these kind of challenges which we have had this study, the massive data sizes, millions of files, etc. So naturally, when 1 million genomes initially we're going to have those all the data, the challenges are huge because we have already 2 years working on this data, and now we are fine-tuning the phenotypes. So I already mentioned that we are saving all the generations. We know all the mating in every generation, in every population over there. But when we are providing for this project up to 4 generation information, so family relationships are really, really important because genes are running in the families. For example, Markus is going to talk about the FinRegistry study, which is the big study in Finland, all the individuals in Finland. So largest pedigree over there is 100,000 individuals. Of course, it's horizontal, but there are family structures on population. And even when you are doing FinGen study, there's 500,000 individuals, there are relatives over there. So somehow that has to be applied in the analysis that there's a family structure in the even the population sample. So how do we then simulate the phenotypes? So they are based on the epidemiological models. We can specify heritability, contribution of the environmental factors, what's the sex and age contribution, smoking prevalence, and other which must be hypothesized, like number of genes, allelic complexity, relative effect size. So we can select any number of SNPs from the genome to have some kind of effect on the trait, plus other parameters. So based on those parameters and genotypes, we simulate the phenotype. And it can be quantitative or qualitative, meaning case control. So we can do QTL threshold models over there as well. So the data flow pipeline looks like this. So we started first with the population simulation, where we simulated all the populations and all the mosaic genomes over there. And then next one is the cut and paste, basically. So you are cutting and pasting from the founder chromosomes those mosaic chromosomes from the bottom generation. So that's massive computational I/O using because you have to copy and pasting and making files and millions of files basically. But then you have to merge those files together so they are more manageable, that they use less space, disk space, etc. But then when we have done all the VCF, we have 1 million individuals in the VCF data format, then we can do the phenotype simulation. Conditional on genotypes and parameters, we simulate the phenotypes. And then when we simulating phenotypes, of course we do the QC and mapping, do quality control for the phenotypes, and then we do linkage and association mapping with the Merlin pseudomarker, doing joint linkage and association analysis, and then we do association analysis with the blink. So we verify that, okay, do we see the signal and how much the sample size must be that you see the signal. So basically we do all the statistics and look, the distribution must be look right, etc., etc. And do we see the signal linkage or LD signal? So currently the simulated genome is, we have more than 1 million mosaic genomes in VCF format. Pedigree structures up to 4 generation. Of course we know everything, but we are providing only 4. Pedigree sizes varies from 3 to up to 244 individuals in one family. Over 42,000 families, and over 200,000 genotyped founders, meaning that they are like unrelated singletons. And I have listed over here 21 phenotypes simulated already, but we have actually more already. So there's quantitative, qualitative, age, sex, smoking status, and then we are now adding new phenotypes based on the polygenic score catalog. So we can take any polygenic trait from the catalog, input that to our simulation, and use those effect sizes and SNPs, which are listed over there, to generate the phenotypes, also using other parameters as well. So that's the, currently we are now fine-tuning the phenotypes, but we will be then transferring this data to the CSC, which is one part of the 1 Million Genomes project, and they will be then distributing that data when they are building the federated system with the 1 Million Genomes project and with the GDI. Yes, thank you.
Thank you very much for this very exciting talk. And as already stated, the questions we will be asking afterwards. But now I would welcome our next speaker, which is Markus Perola. He's a research professor at the Finnish Institute of Health and Welfare, and he will tell us about FinRegistry, a population-wide big data registry study utilizing AI for disease trajectory prediction. So please welcome.
Thank you. It's a pleasure to be here. So I'm talking about, Tero talked about simulated data. This is truly real data, which is updated every year, and it includes all the people in Finland, or all the residents of Finland that have the social security number. So if you have a social security number from Finland, I thank you for participating in this study. So what have we done? Our institute, Finnish Institute for Health and Welfare, it's our law-abiding duty to study and follow the health and welfare of Finnish people. It's in the law considering us. So we have thought of doing this now the big and the right way. We have collected together 19 different health-based registers in Finland, which cover many aspects from health to socioeconomic status, to salaries, to benefits, etc. And we use that to form this great data set of 7.1 million people. You might wonder what's happening when in Finland there's only 5.5 million people, but we have included all the people who have been alive in 1st of January 2010 and their relatives who are either deceased or who have born after that for them children, which have registry data. So that becomes the number from. And currently we have almost 7 billion lines of data, and the lines have many data points, so we have billions of data points in this big data set. And we use AI, mainly machine learning, neural networks for disease outcome-specific prediction models. This I will give you some examples what kind of work we have done and what can we do done with this great data set. Then another important point is that we also get like an online prevalence of cross-sectoral prevalence of almost all the ACDs in the ICD-10 book at present, and we have a specific website for that that's open for all called Risteys. This is actually a collaboration with FIM, which is the Institute for Molecular Medicine Finland, and Dr. Andrea from there, and his team has been working us together from the beginning, and we also work together with FinGen project. So we use this nationwide registry to build machine learning models that will help to understand better. So it's kind of a hypothesis-free approach. My background is in genetic epidemiology, but I have done registry studies all my research career. And here we use genetics mainly in the pedigree structures, this kind of old school genetic epidemiology Tero mentioned about. So we have also information on the registers about the family structures, which we can use to do heritability analysis and these kind of things. And and also model the families into the disease prediction models. I will show you some examples about that. So if you go to the FinRegistry.fi, you can find this kind of a picture where you can click on these and you can actually see our data dictionaries, what kind of data we do have under each of these 19 registers. So please go there, and there's more information about the study as well. Of course, this is very sensitive data. All the systems that we use have been externally audited. We have an ethical positive statement for the study, and also we communicate with the citizens who can exceptionally actually withdraw themselves from the study if they want. But we have very few people who have done that, less than 5. We have an open website, both the THL and FIM, actually, for this, mirroring each other. So what do we do? So as I said, we do disease trajectories in the Finnish population and also the prevalence and in the population. I want to stress that that's cross-sectional, not lifetime prevalence, because there's a distinct difference in the usage of each. But for example, now any physician in Finland can go to the Risteys website and check what's the prevalence of his or her favorite disease in Finland at the current moment, at least according to the registry data, if you believe that. So we cross-reference the registries to each other. We are just not using them like one at a time. Actually, one of our biggest files in these 6 billion lines is the combined endpoint dataset, where we have used several different registers to form these disease endpoints. So we don't just trust one register of medicine or say disease hospital discharge register, but we combine and make certain demands that a certain outcome is reliable. Actually, Tero here has programmed a so-called endpointer that helps us to do that. So Tero works here as well. Endpointer is actually available as well if you ask from Tero. We have processed the data. This is an old slide. Now more than 6 billion rows. We got 2 billion rows last, actually June. We got all the laboratory results done in Finland from 2014 on. Actually, the data set is complete from 2021, but if you have gone to a doctor in Finland and got your cholesterol measured, we have it. What we have done with this is that there's now a couple of examples of published or soon-to-be published results. This first paper is now in press in Lancet Digital Health. We wanted to use this setting, remember, 1st of January 2010. So we had a lot of children born into the cohort or who were children when we founded this. And the RS virus infection is a very serious infection, often for infants, young children, and we collected a cohort of 1.2 million Finnish infants from this cohort that we followed up for until up one year. So of course there was a lot of them because we have all the Finns. And the outcome was whether you were hospitalized for respiratory syncytial virus. And there were 17,000 such children. And then we did the machine learning trick. So we asked the algorithm to tell us which associate with the disease. And here on the left side you see other parent, and on the downside you see the mother and father was on the y-axis. And as you can see, there are different risk factors derivating from the parental phenotypes for the child to get the disease. I'll show you more closely this in the following slide. So there are actually some parental risk factors, and this now brings back to the value of the family information in this data. So we saw that there are some of immune-related, like allergies, urticaria, asthma. Those are probably genetic risk factors. Then there are some gynecologic obstetric. So there was some, if you have problems with lactation, for example, that's probably multifactorial. And then psychiatric of the parent. So that might be like truly external reason for a child to get hospitalized. Maybe her or his parent is too occupied with her own health, or his own health, so they can't, really manage with the sick child at home, and they have to turn to the hospital. And same goes probably with the parent's substance use. But of course, there was specific risk factors for kids as well. A number of young siblings. Well, maybe you are more exposed when you have a lot of sisters. Congenital malformations, that's quite obvious. Some pregnancy-related things. So if you're, in trouble from the start, and then many birth complications, and all these will be out in October 24, so you'll know more then. Then another story, we started this already before the COVID, but then during the COVID we realized that this could be done, we could done something COVID related with this. So we started this study on who takes vaccination and how we can define those groups in our nation who don't get the COVID vaccination, so we could maybe target, advertisements to them. That's in the core of what DHL should do, I think. And this is just a study structure. We again collected data from all Finns for the first vaccination, because that we thought that is the most unbiased, and we excluded the young adults because they were excluded from the vaccination when there was not enough of them for a while. So it was adults from 35 and so, and the most important predictors can be seen in these figures. So actually, salary was one of the most important, but also whether you were born in Finland or not, and other things as well, like your city of residence and so on. So this is all published in Nature Human Behavior. This is just another way of presenting these. We have some of the cancers here, and some of the cancer medications, actually, or immune medications that we're exposed for, and we can see actually some of these municipalities where the vaccination was very much advocated against in these, for example. So this really told us, as an institute, how you could really target your vaccination programs, and this is now published in Nature Human Behavior. This is not even submitted yet, so this is very secret data now. But anyway, I trust you. We're following. We analyzed mortality of the following year. Can we predict that from registers? Actually, the first author will be Latvian, so good for you. And Latvia, but he's a very good statistician and works for us. And here you can see the very first results of what we can see. So at the top, where the prediction is least, are the accidents and other things that have no really predictive factors in the registry, and down we have like Alzheimer's, and actually COVID was pretty much predictable from the registry data. What was interesting but not surprising was that the young people their deaths were most predictable. Why I say it's not surprising is because death in young people is very rare and usually has either a disease in background or say that drug abuse or otherwise not recommendable behavior status. And we also look which data matters in our prediction of the 1-year mortality, and really the hospital records from were the most predictive, not surprisingly, but they're much more predictable at the end than the socio-demographic features. But the socio-demographic features, of course, had also meaning. Then I wanted to just show this. This is Risteys. I'm sorry, this is Sol, but if you go to the risteys.finregistry.fi, you can try it. It's in English, so you can try our prevalence data. And there is a certain structure. I advise you to read the instructions before you start. You can start playing. I don't deny that, but you get more if you read the instructions as what is what, and we get less questions asked. Then I just wanted to advocate. We haven't yet published anything on this, but we have this exciting, I mentioned in the beginning, the Kanta, so which is our electronic health record system, national one, and or health register system, and we have, as I said, all the companies, all the hospitals, primary health centers, all the laboratories that do laboratory analysis in Finland, they have to put their data into the Kanta system. And we got the data this summer. And actually we got it first time in the fall, but now we got a new version of it. And we see that most individuals have at least one measurement in the past 6 years, so this is pretty filling. And actually we had an average of 130 lab measurements per individual, which is quite a lot. Of course, there were people who had none. Actually, very small children and then aged people had the most usually, or the most have, and on average, so of these 360, 80 were unique. Of course, you are worried about the variability between the laboratories, but they are very well standardized in Finland, as we can see from the glomerular filtration rate in this picture. The distributions in the sample of laboratories follow well each other. So this is very exciting data. We are now looking for imputing the data for people who don't have, for example, creatinine measured, whether we can use that. And for example, we have identified that almost 18% of people who have creatinine values indicating renal failure don't have that diagnosis. So either there's a problem with the registers or in the diagnosis. We don't know yet, but we will look into that. So there's a lot of hidden data in here. So we have a lot of data that's not usually available. We have all the family information. We have living histories about whether you live in assisted facilities, which is sometimes an important endpoint, and welfare benefits. We have drug prescription information, also for non-reimbursed drugs and intensive care information. And in the near future, we will move this into because we are now not under the Finnish law for secondary data use, because what I told you in the beginning, it's our duty to do this. But we will move this to other Finnish researchers and also to you if you want to study. You have to apply through FinData. We are in a process of transferring the data to them, and Tero has a big role in there as well. But anyway, the FinRegistry will continue in TSL as planned, but we just want to we think that there's too much data for us, a small group, analyze alone. We want you all to join us in this analysis of this wonderful data set. Thank you.
Thank you very much. Indeed, a very valuable resource for precision medicine. The questions then please save for later. But now we move to Latvia and we move to image analysis and beyond. I would like to ask Maija Radzina. She's an associate professor and researcher in radiology at the University of Latvia and Riga Stradins University. And her talk will be on AI in imaging, a threat or opportunity. Please welcome.
Thank you, BAIBA, and hello, everyone. My talk will be less about humongous amounts of data, more about clinical use of those data and maybe some of the opportunities that we should look into and maybe some threats that we should think about before we move on. So I will start with a quote from Stephen Hawking, who said that this will be a spell, almost for the end of the human race if we will work on the artificial intelligence coming into our lives. Actually, partly in certain fields we already observe this phenomenon. Also it is in a chess play, and also we know that the cars are driven by AI solutions and we don't need the personnel anymore. And also in my field that I represent, radiology and imaging, it is already in some of the entities proven that machine finds some irregularities, some problems faster, it can mark it. And in the way, when you compare the person performance, we may not be as fast at some moments, especially when we are more tired or overwhelmed with the flow of the work. And this is very true. And I was astonished that these were data from 2018, published from UK, with increasing workload and shortage of radiologists. And this crisis have reached us in 2021, just after the COVID. And we are quite deep in this workforce shortage, especially in imaging, because as you can see in the chart, that the computed tomography and MR scanning globally is rising substantially and by every year. But the workforce is not following those digits. So more or less, we work every day more. And as you can see in that picture, the work comes to your house with a crying toddler, and there is high probability that some findings and some little nodule somewhere might be missed in such a setting. So there is one of the components that we think that the AI may maybe replace some of us or may replace some of our tasks in the basic needs. So of course there's a lot of research on where we could apply the AI in radiology. And as we know, AI, it consists of the several sub-layers of it, including machine learning, deep learning, and actually it has been there for 20, 30 years. So radiology actually is one of the most digitally informed healthcare segment already for decades, and we do not feel that we are now introduced recently with this. And let me show you what could be the most typical impact in our imaging with incoming more and more solutions. So one of them is basic detection of something different. So computer-assisted detection systems, and this includes also maybe the labeling of the findings. So I don't need to count things. I don't need maybe to measure things which are quite standardized. This could be automated, of course. And also it can help my workflow by prioritizing certain imaging findings, bringing them more to the priority in my long list. Or maybe this risk stratification and prognosis can be elaborated in some percentage by saying, like you showed from these genomic data or other causes, that your likelihood of the mortality, depending on this and this, is with this likelihood. So what is this hype versus reality? So AI, if it will replace one part of the doctors, there is high probability it will replace all the doctors. And I will tell you why. There is no medical field that is not relevant or not related or not dependent on radiology. Any diagnosis that is done includes the certain imaging data into a certain time point. So reality actually is that we need the radiologists increasing numbers globally who will use this tool. So why it is so important? Let's take a look at this quite simple pyramid. So number one is the technical efficacy, which might come from AI or maybe come from technology itself. Then we may provide the diagnostic efficacy. Then we will see the diagnostic impact on our healthcare or maybe on each individual. Then we can decide the therapeutic pathways and observe this impact. And then there is this outcome that we want to measure as a value when we talk about the value-based healthcare systems. And then we can measure the final is socioeconomic benefit. So somehow diagnostic part is quite at the bottom of that pyramid. So we cannot omit this truth. So how to enhance this value-based radiology? And there is certain activities with this. Number one, we need the correct question. It is referral information, and this should be structured. And I think all of us are really artists in medical field, but the questions can be asked in more standardized ways. So then we will be able to evaluate, is this examination really needed? And does it follow the certain guidelines and certain protocols? And then we can also standardize how these patients will get to the examination rather than being chaotically in the big pool and fighting for their examination. But then they will be targeted in a specific pathways according to the first question. There is also initiative from EU point of view when we want to justify and to avoid unnecessary CT and MR imaging. So this is the project called EU Just CT. So we really need a good reason to do the harm to the patient, to apply this ionizing radiation, because the examination has the impact only when there will be change of the concept or change of the disease, change of the diagnosis. There should be a certain value of that examination, not just have it. So for the common standards, we need the common language, which can be defined by different shortage names like RADLEX, SNOMED, and the others. But the point is that this also cannot be applied only to one institution, to one country. So we really need to start to talk the common language, or at least the common coding. AI in radiology nowadays is expected to have an impact on each person's daily routine, like me, radiologist, in, as I said, image acquisition and also processing improvements. So there will be maybe certain added values. There will be more colors, there will be more labeling, there will be improved scanning time. So I will gather more data in a shorter time, but these more data probably and hopefully will be partly processed by AI. And I already will get some hybrid mid result that I will continue to post-process with my human intelligence. So only then I will be able to prioritize the patients. And also it's important that all those data are really collected somewhere, that they are not chaotically, but they are also classified, clustered, and then we can analyze what is the prognosis, what is the prediction, then we can work on the prevention and tailor our behavior. This is a simple example how we expect as a radiologist, how AI can help us. So yes, we want this color coding. I want things to be labeled like, I don't know, mark me gray everything that is not so important and mark me with a color something that I really need to look at. And this is what is already happening. So a lot of pathologies are already not only labeled, so the segmentation itself is one of the basic solutions. We already want AI to move further and to help us in characterizing, at least maybe in two folds, like it's malignant, benign, or maybe it's treatable, non-treatable, so it has enlarged or not. So there are different types of these algorithms that can be used, and usually based on convolutional network calculations. So then we can classify it, because a lot of things look alike, and AI is learning only on already labeled data sets. And the fact that we can buy some software from somewhere does not mean it will be effective and good on our cohort. So this is the common and very complicated task to teach the right things in the right way also to the AI, rather than just relying on it. So what is important that from very large data sets, it also can move onto the artificial part. And artificial part, it means it can build up the data that are not original, that are not mine. And this is what is happening. So I'm actually scanning something really different, and it enhances, it changes, it reduces something, but it builds like a bit artificial reality. Yeah. So it can build me the lesion where it wasn't, and it can remove me the lesion where it was. So this is the one of the big threats that we should look into that. We don't know what kind of program, what kind of algorithms can be embedded in those AI programs as well. So this is something that we should think about. So these are just few examples. So the Baltic states are also among the many countries which work on solutions. So this is just one example about how the organs can be segmented, meaning colored and marked in a colorful way. This is also the solutions from Lithuania, detection of the lesions in lungs and some automated measurements for echocardiography. Latvia works on a stroke and detection of the lesions in the brain. So each of the country in many hubs are trying to find what is beneficial for the clinical community rather than just developing the algorithms. So what type of scenario and how does the European radiologists, the professionals who are overwhelmed with that workload, look at the AI? There was a survey, and it was addressed with a way that, unfortunately, primary interpretation with replacing the radiologist can be really poorly supported, only by 7%. And actually, most of radiologists see this as an assisting tool. And we are very much looking at this lower blue bar, that we want AI to improve our workflow. Yeah, this is what we actually need. Also, there has been a very interesting publications about educational potential. But here we also, again, run into some risks. What is the risk? Because when we calculate, we say that radiologist itself performs on average about 80-something% sensitivity and specificity of finding something. Trainee, resident, also not so great numbers, even lower than the certified specialist. But when we add the AI to this, then everybody will perform better. But this is not about replacing the board-certified specialist by resident plus AI. This is about replacing maybe my time that I can dedicate on more sophisticated and difficult situations and cases and clinical scenarios. And the teaching part, basic teaching part, maybe could be delegated to the AI. But then the question is, where is the ground truth? And has AI taught my younger doctor the correct, true findings? First machines will do a lot of jobs. Yeah, we should be positive, but we should manage it well. Otherwise, it will become a concern, right? With Bill Gates' words. So there has been a quite worrisome text about influence on breast imaging and how do we report this and how do these findings come to the patient who are involved in screening programs and how do they receive their results. Also, we used our AI solutions that were available globally at that time, at the COVID period. And unfortunately, our results said that correct grading of COVID findings and severity of the lesions were with the AI precision only by 40%, not even 50, which is lower than just throwing a coin. So we cannot rely only on AI. Also, multitasking is a challenge for AI protocols. This is that capacity of the human intelligence to do the multitasking, to do the multifactorial, to do the multi-layer analysis. Otherwise, it doesn't make sense and we cannot really answer the complicated questions. So then raises the question, so why do radiologists don't use the AI? And they don't use the AI for the main reason. They don't see the additional value yet. It doesn't mean that we are against. We just don't see that it outperforms or performs not so well. And for some of the survey respondents said, it adds me even more time, so I need to work for it. I need to label it. I don't want to do it. So there is another guy who said that we are summoning the demon with having artificial intelligence. It would be interesting to ask what he thinks in maybe 10 years time on. So these concerns are, of course, addressed by a lot of legislative aspects, and we should think about the data safety and good governance of it, and also accountability. Who is responsible of data, that they are not leaked, that they are correctly performed, and what if they are interpreted wrong? Yeah, who is responsible? And there are a lot of regulations that didn't put all of the white paper, which also came out, but the point is there at the bottom, and specifically it says that number 3, ensuring that AI works for people. Not that we are adjusting, of course we somehow adjust our life to the presence of AI, but it's working for people rather than we are adjusting our lives for it. So would we be happy to receive this automated image interpretation saying that the probability that this lesion is abscess or probability that this lesion is cyst is 0.7 something, 0.12 something. So how does the patient read this? What does the doctor can draw out of this? And instead of having like it is a cyst and that's it, or it is the abscess or not abscess. Will we be the radiologist sitting at the desk just clicking the accept or reject? I don't like what AI proposed. Maybe I want to change something, yeah? Have there been any major problems with integration of this, and what does the patients know about? Have their data being processed by AI software? And you see that actually most of the European radiology institutions said, unfortunately, patients were not informed that AI software have worked on their data in 75%. So there are potential concerns, and what is important that there should be a guiding principles, how we spread this technology and how we evaluate what is valid. And there was an interesting, and this will come out very soon, there are purchasing considerations. Before we purchase them, what is main problem solution which will be addressed by this AI solution? How can AI performance be monitored? How will I address the quality, and what is actually direct and what are indirect costs of implementing this exact software? And there are just a couple of minor last comments that AI systems actually have bias. Bias by producer, bias maybe of the, shareholders, co-CEOs of those companies, including some of the radiologists are also as the founders of those companies. So why my software is better just because I'm a radiologist? Maybe yes, maybe not. And we need to also firstly meet the clinical needs rather than just performing the development of the protocols. And it should promote the well-being, and we should not harm the people just for the sake of having it. So we are the inspiration for all of this development rather than this AI solution is inspiring us. So we must remember that we should be the ones who says, I need this software for this, and please develop this. And then this and that will benefit from that. So then we have to come together, educators, researchers, stakeholders, the business people, IT people, and do the very good networking. And this is where this multi-layer comes in, and then we can come to this conclusion that it is a tool, not a threat, if it is well governed and we know the risks. And I'm actually looking forward to those solutions that will surprise us in the near future and will help us to learn. Thank you.
So thank you very much for raising indeed these very important real-world questions, and I hope we will have a chance indeed to discuss this after the session. And now I would like to welcome our last speaker. It's Andrea Townsend-Nicholson. She's a professor at the University College London in UK. And she's also the training lead of CompBioMed, which is a European Commission Horizon 2020 funded Center for Excellence in Computational Biomedicine. And her talk will be on CompBioMed and the virtual human, building digital twins for healthcare. So please welcome.
Thank you very much. It's a real pleasure to be invited to speak here. And this is the successful outcome of a collaboration between the CompBioMed Center of Excellence and two of the EuroCC National Competence Centers in HPC, NCC Netherlands and NCC Latvia. So, a quick disclosure so you know my background, where I come from, and where my conscious and unconscious biases may lie. I'd like to introduce you to the virtual human. This is a 6-minute film, and it will encapsulate everything that we are working towards and hope to bring to the clinic.
Our bodies are exquisitely complex machines. In our efforts to care and improve them, we look to those who push the limits of what is humanly possible, such as athletes. Now, you would say, what can we learn from them if every athlete is different? If they achieve peak performance through customized diets, training, and technology. As it turns out, we too are all different. That is to say, there is no single lifestyle, no diet, no medication that will work equally well for all of us. If this is so, how can we get the best out of our bodies? Now, imagine a virtual human, not made of flesh and bone, one made of bits and bytes. And not just any human, but a virtual version of you, accurate at every scale, from the way your heart beats down to the letters of your DNA code. You know that many drugs only work well on some people and can cause serious side effects in others. The reason is variations in DNA, our genetic differences. But we understand how these DNA differences change the building blocks of your body, the proteins, and we can simulate in a computer how drugs interact with them. By testing drugs on your virtual body, your doctor may eventually be able to test a wide range of drugs and select precisely the right one to suit you. But sometimes, choosing the right drug is not enough. We must also be able to deliver it to a precise target in the body. Take inhaled drugs, for instance, which can end up in the walls of your nose or at the bottom of your lungs, depending on the intricacies of your particular respiratory system. Using supercomputer simulations based on scans of your lungs, we can predict with high precision where particles will flow, and then design devices that can deposit drugs exactly where your body needs them. Well, obviously, mastering drug delivery means controlling the body's main transport network, your circulatory system. By simulating the movement of red blood cells and other cells, we can understand important protective processes, like those that prevent blood loss after an injury. Virtual humans could help doctors to plan risky surgery too. They could be used to work out how to reach an aneurysm deep in the brain that is at risk of rupture, which could cause a stroke. Surgeons could then try out the best treatment or implant to suit the location and shape of that particular aneurysm. They could even double-check that the implant will not cause problems circulatory system to drive the most remarkable pump that evolution has ever created, the heart. To create a virtual heart, processes have to be modeled at multiple scales, from the contraction of muscle to the blood flowing through its chambers, to the movement of charged atoms during a contraction. Virtual hearts already beat within supercomputers, where we can test the effect of different drugs or pacemakers, but also more fundamentally, to understand the way an individual's heart works. Your virtual avatar will not only be built in your image, it will also move like you. We can use it to calculate the forces and the mechanical stresses that are constantly induced on your bones, and predict the risk you will suffer a fracture. Doctors can also use virtual humans to work out how diet, exercise, or drugs can help those with brittle bones. And what if we go even further than treating disease, using virtual humans to prevent illness? Within a supercomputer, many virtual versions of you can explore small changes in your lifestyle and how they affect your health, aging, and quality of life. Virtual humans, helping you to figure out the very best that you can be.
So this is our goal, to build and to bring this technology to the clinic. And in a sense, the virtual human is actually a digital twin of digital twin components, and we're increasingly building those. And the approach to it is to combine data and knowledge from multiple sources. We're working from the atomic level of the nucleotides within the genome, all the way up to the macromolecular structure of the architecture of your skeleton, for example, and taking all kinds of different forms of data from integration of mechanics, imaging, cell models, and all of the omics, and putting them together. So in this first example of building, this is very relevant to molecular medicine and drug discovery. We use variants within people's genomes to understand the changes that that elicits in target proteins that can then be used for calculations of binding affinities with drugs, to understand within a patient population or a global population, what is the best therapeutic treatment for individuals. And this is treating the individuals as individuals with this methodology. This is another example of taking a look at putting two digital twins together. Ultimately, all would be coupled together in the virtual human. This is looking at two codes. One is LIAA, which is the heart simulation model that comes from the Barcelona Supercomputing Center that is generated by taking CT data, so imaging data are critical, and also EKG data, and putting those together. And another code called HEME-LB, which looks at blood flow through the human vasculature. And by coupling the virtual vasculature with the virtual heart, you now have a virtual heart that pumps blood through this virtual digital twin. So how do you bring these into clinical practice? And this is where we come into difficulties, and why I pivoted very quickly from being intensely interested in my own particular area of research into being much more interested in education and training. Because if you look in this graph, these are data taken in 2016. Archer was one of the UK's national supercomputers. Cartesius is now out of service, but was the Dutch national supercomputer. And BSC, most of this is Mare Nostrum, but some of the other Spanish Rizzo's machines. You can see that the use of supercomputers in 2016 by Life and Health Sciences is incredibly low in all these areas. And in fact, only 0.2% of what was running on Archer was medical, and most of that was medical physics. The rest was things like molecular dynamics, simulations of proteins. So life and medical sciences are a disadvantaged demographic on computers. They're not used to using these in their practice. They're not used to having access to the machines. And so we set about changing this. I'm just going to put all these elements together for you. I was running the biochemistry degree program at UCL for about 11 years. So I was in a unique position of sitting in the center of excellence that was aimed at building digital twins and running a teaching program where I controlled what went into the curriculum, which is an incredibly useful thing to be able to do because we could slot these things in. If you have not got access to resource, the compute resource you need to use has to be relatively small because the more students you put on it, the more compute you're going to consume. And microbiomes and their role in health and disease was becoming increasingly topical, so we picked a medically relevant exemplar, and we went through a whole methodology of purifying the microbiome, sequencing this information from the right hand of the skin. This is a handshake experiment. If you meet somebody, you shake with your right hand. So we collected these samples. There is no problem with personal data or ethics. And we went through a workflow where we worked out what was in these sequences and whether there were correlations with particular factors in the metadata. And what you can see below, and I'm very pleased to see Illumina as one of the sponsors here, is the only, at the time, and I would like to find out if it still is, I-Seq NGS facility in an undergraduate university curriculum. So, starting in 2017 until today, we have been teaching medicine students, we have been teaching biosciences students, we've been running extracurricular courses through the PRAISE Advanced Training courses. And as you can see, we're learning how to teach better and faster. And this is one of the key elements that we've learned from our colleagues on the computational side is automate and scale, automate and scale. And this is what we've been doing to the extent where we are now testing, teaching 300 students at a time how to use these resources. And we are porting our courses to other universities. So I teach this at UCL. I also teach this at the University of Sheffield, where I'm not a member of staff, and I co-teach it with another individual, and we are using these opportunities as test beds to work out how to make these training resources available to other people if they need to be in the curriculum or not. It's also really, really important to engage with everybody in the ecosystem, from the policymakers down to the public. Both of these categories will have people who are patients within them and have a vested interest in understanding more and having a voice. So on the left, you'll see the gentleman who is the second from the left is a member of parliament with particular interest in AI technologies and other assistive technologies that can be integrated into healthcare to improve health and well-being. On the right, we go out to public engagement activities all the time, and if you see people writing on each other's backs, it's because we give them a questionnaire and we ask them, would you want your doctor to use the virtual human to treat you? Would you want them to use it to predict what you might have? And would you use it for yourself, for example, with fitness tracker data to manage your own health and well-being? We're particularly interested in the free text fields, which reinforce people's wish that data be made freely, accessibly available, and that they be able to derive benefit from the use, and that there will be no social stratification in access to these resources. So our plans for the future, in the same way that we started the training courses within the curriculum at UCL and then ported them outwards, we are assembling a UK-wide medical school network with the intention of expanding that into the European network. Once we figure out how to do it with a relatively contained number of medical schools, then we are going to be coming to you, and we look forward to this opportunity. We're creating summer and winter schools because not everybody can put this sort of thing into their medical curriculum. We could, but you can't, for example, in the United States. We are training the trainers. I have to train with someone else at Sheffield because I can't find the me at Sheffield who can do the whole thing. So we're going through this, train the trainers and then unleash them in their own areas. We're surfacing in Sheffield what is their particular area of research relevance for medical students and building a course there and bringing it back to UCL. So we're starting to explore portability of courses. We are always up for finding training buddies and pooling our resources. And we are in the process of creating an international CompBioMed society so that we have a one-stop shop for conferences, for knowledge exchange, for training materials, and for finding ways of allowing people, including patients, to have a voice in the creation of the virtual human. And I just want to acknowledge the ton of people. It's a large slide, probably very difficult to read at speed, but I believe it will be captured in the recording. And we work in an ecosystem. We need everybody from all sorts of different dimensions in the HPC facilities, clinicians, patients, basic scientists. Everybody has a role and a place, and a huge number of people helped with this. If you'd like to know more, these are our social media handles, and if you are particularly interested in what is underneath the hood on building the virtual human, the coordinator of CompBioMed, Peter Coveney, and colleague Roger Highfield, who's one of the directors of the London Science Museum, have written a book on Virtual U that does go through much of the physics-based modeling and the history of how we came to be here. And I'd like to thank you very much for your attention.
Thank you very much for letting us see the future of personalized medicine. And now, indeed, thank you very much for all the excellent talks. And now I would like to ask all the four speakers here to take the chairs and the microphones. And we will be happy to take some questions from the audience. And of course, the speakers are welcome to ask each other a question as well. So here is the microphone. And perhaps, indeed, let's start with the first speaker. Do we have any questions for Tero Hiekkalinna? So his talk was on the 1 Million Genomes project, simulated synthetic genomes. We have a question, please. We have also some mobile microphones, so maybe.
So, thank you. It was a very interesting talk. I was curious, when you were simulating and splitting those genomes, did you use the data from the HapMap? You know, the one that tells about the linkage disequilibrium?
Yes, we use that data, those sequence data from the consortium website, yes. And we use those 5 selected population, we put those as a founding population, and we scramble the chromosomes over there, and then we are using those constructing the mosaic genome. So HapMap, the original HapMap, we are using that data, yes.
Oh, thanks. It was good. Yeah.
Thank you. I think we could take at least one more question. Any questions from the audience? So otherwise, maybe I would take myself the liberty to ask, because indeed, sorry.
Hi there. I just wanted to ask a quick question. There's increasing evidence that there's a lot of circadian rhythmicity and changes through time on how we express genes and how phenotypes change throughout the day and through different seasons. And I'll be very interested to find out if you have any thoughts about that. By the way, all of your talks were fascinating, but I was wondering the changes in time on how we observe phenotypes and gene expressions are very interesting to observe in your databases and potentially in the virtual twin, and if that's something that you have considered in your research.
We haven't considered any kind of seasonal changes in a phenotype in simulation, but I guess in Finland's study there's some phenotypes which they are analyzing as a seasonal changes as well. If you, Markus.
Yes, and in both FinGen and FinRegistry we are doing that, but of course Tero can simulate whatever you tell him to do.
Sure, we can simulate those kind of seasonal effects if we wish, but we don't do that, yeah.
Thank you. Yeah, indeed. My own question would have been similar because, of course, we're simulating the genomes, but all this the phenotype is much more complex. We have that environment and lifestyle factors, sometimes unknown, hidden confounders. Do you have some mathematical way to build in this uncertainty, how you somehow see how your model might deviate from the real world, so to say?
Sure, we don't know what is happening in the real world. We are trying to simulate and mimic that. And of course, we are adding uncertainty by adding the random environment variation for the trait as well. But of course, we try to mimic the reality and think about those parameters when we are simulating the phenotypes that they represent somehow reality. But of course, we don't know everything, but we try to do that.
Thank you. Now, indeed, perhaps let's move to the second speaker, Professor Markus Perola. Do we have any questions related to FinRegistry? Please.
Yeah, thanks for the presentation, Markus from Riga. Actually, I have two questions. First of all, very simple, whether also the Finnish cancer screening registry is linked to this data registry system. And then the other one, what about the legal rights, for instance, in the case if you can predict a case of death in a young age individual, can you come back to prevent this?
Yeah, thanks. Good questions, and especially in that sense that I can answer them. So the second question, the Finnish law explicitly says that we cannot use registry data now for such a purpose. So it's kind of a sad thing for us, but it also makes it easy because there's a specific saying that using registry data, we may not contact a person. That's probably in the law because it wants to protect the privacy of people. However, in our very far future is the wish that we could use these models actually, like a GP could use registry data in making preventive decisions about his or her patient. So, but this would need law changes actually. But this is one of the reasons we do this work, that eventually this would be found useful and could be even found so useful that it could be used in the everyday work of the GP. And what was your first question, sorry?
About the cancer screening registry, but the question is also linked to the previous one, because if you don't have the rights to use this to intervene, then probably you cannot use it also for designing personalized screening in this respect.
Yes, of course we can use this as a model for any kind of a personalized screening, but we cannot go to a person that say that you are now in risk for this. Yet, we hope that in future we can. But yes, we do include the Finnish Cancer Registry and the cancer screening programs to this dataset.
Thank you. I guess we could take at least one more question from the audience or the other speakers, maybe.
I have a question. When you were showing the data about the paternal and the maternal disease impact on RSV in infants, it looked like there might be things in the paternal side where there was actually a positive benefit. Did you identify things like that, and are they informative for?
Yeah. So yes. So I consider myself a risk always, two sides of a coin. So you can put it. You can working. You can do it both ways, but yes, there were some protective. I didn't show all the data, but some protective in both mother and father. Yeah.
Thank you. And now we move to Professor Maija Radzina. So do we have some questions from the audience related to imaging and AI? So please.
Hello, thank you all. Very interesting presentations. I wanted to ask this, how available is for general public? For example, I do mammogram and I want to check myself with some AI tool. Is it available these days?
It's a wonderful question. Thank you.
I think that we are quite far from this in terms of cancer diagnosis. So I think everyone already is at least have tried to measure the blood pressure, and we have a lot of those registering smart watches on our wrists, and it counts a lot of things. But I think to the terms of, is this a normal tissue? Is this maybe something wrong? We probably may get to automated systems that may help you to get the data. But interpretation will always have some, let's say, obscured part, because all of us are quite emotional and sensitive people when someone tells something about even how we look, just phenotypically, right? So what if we will start to rely on just automated program that will say there is something worrisome in this test, you should see the doctor. This even very vague and very open phrase will keep you so anxious until you will get to that doctor, that in these situations, so far the system, knowing the psychology of the worried patient, wants to prevent that psychological harm of worrisome waiting, which is very hard to maintain on your own. And this is the burden of nowadays digitalization, that a lot of patients already in advance receive the reports, receive the conclusions, they receive also some histology results, and they do not know exactly. Then they read a lot of internet materials and they make wrongful conclusions, and it is not always beneficial. It really depends on the intelligence and maybe the analytical and background information, and also background mood for those patients. So this, I think, we are not yet there for sure. For some basic things we might be, but like, I don't know, likelihood of your headache, or I don't know, measuring your some other phenotypic, or maybe some temperature and body data telling you when your menstruation cycle gonna start or so, probably these will come into the life. But I think cancer is really important and life-threatening condition to be relied only on some printed information on the screen. But thank you for question, it's wonderful.
Thank you. More questions from the audience or the speakers, maybe?
I actually have one.
Okay, please.
What do I need to do or see put into a medical curriculum for students to help radiographers be more comfortable about accepting AI methods? What sorts of things could we teach that would give maybe greater confidence, or make it easier to interpret, or perhaps develop a culture of a pipeline or a workflow? What could we do to help?
I just want to remind that we already use it, we already have it, and there is no excitement in any imaging and radiology community in all the levels about the existence of these programs. The trick is that we need more, maybe those algorithms, and maybe some of the programming and some of the IT, quite sophisticated, but still basic knowledge included in some medical studies as early as possible. Then this will be better taken. Also, as soon as we show where this can be applied and why it is useful to learn it. Not just to give it an optional course, but to try to move it in mandatory level. Because AI is everywhere. It's not only for medical students. I think it should be implemented in any of the educational systems. Why? If you are knowledgeable, if you know what you are facing, how it works, then you will be critical about the things that are coming out, about the results, and also implementation and use later. So it's information in advance, which will tailor as they grow. So that's what I think,
Okay, thank you. And so now we move to the last speaker. Any questions for Professor Townsend-Nicholson on the virtual human, please?
Yeah, I actually have a question. So my name is Annette. I'm from okay.
Gotcha.
I'm from Riga, Stradins University, and thank you, first of all, for the fascinating talk. And I have more of a question regarding different approaches on how to teach computational bioscience and computational medicine to students. So the thing that I have seen is that there are many PhD programs that have appeared that the teaching staff is mainly based in AI and does not have, say, the necessary maybe bioscience or medicine background. And what do you think of this kind of approach when it's taught by AI people, not the medical science background people? And how does that make an impact on our future leaders in medicine and whether the results are going to be interpreted the same?
That's an excellent question. I think one of the problems we have when we start to say we need to do things in a multidisciplinary manner is that we discover that there's no person who's dually fluent in the two components. So what you're looking at in this particular area is taking a field that's been primarily experimental and less computational or clinical, if you want to look from the medical side, and starting to integrate clinical methods into it. So it's obviously going to be most engaging for people if it aligns most closely with the primary thing that they wanted to study anyway. And it's slowly happening, and I think in 3 or 5 years, we'll have a very different conversation. I'm an experimental molecular biologist. In the last 7 years, my work's gone from being 98% experimental to 95% computational. And we still do experiments, but we work out very carefully what we'll do. So we make better, more clever use and higher throughput technologies. And the thing that was the game changer was the availability of the atomic structures of the G protein coupled receptor proteins that I'm interested in, and the fact that open source software was available that allowed us to do the kinds of compute we wanted. And then we hit sort of a firewall when we discovered that most of us couldn't get near a supercomputer. So I think doing this twinned teaching, and I chose to do it at the undergraduate level because I think PhDs are too late. I think masters are too late. And in the European Commission system, we've had large conversations about how we can do this, and what turned out to be a unique feature of what CompBioMed was doing it at the undergraduate level, because that's controlled by individual universities, and it is very, very difficult to get a cohesive, coherent program together. I mean, in any discipline, whereas you can have masters across multiple universities or PhDs. So I think it will change. And I think that what will happen is there will be a slight displacement of the AI experts and the HPC experts who come from more physical science backgrounds and are working in the life sciences as the life and medical scientists start to move into the area, understand it better, start to train the trainers, teach each other, and move forward. But in 5 years, it'll look very different.
Yeah, thank you very much. I'm going to catch you later to discuss that.
No problem.
Thank you. One last question in this session, still possible?
I have a very short question about this virtual twin. How far we are and what kind of data set you need to do what you are able to do now?
It's a good question. In the cardiovascular area, it's mature and it could go into the clinic now if there was a regulatory pathway. In some of the other areas, for example, the neuromusculoskeletal one, there's a facility in the UK where GPs or radiographers can upload the data and have the risk fracture prediction calculated. The integration of everything together is a little bit longer, and building new digital twin components, we are focused on the 3 you saw, but are looking for others. So one of the joys of coming somewhere like here is finding people with data sets that they're just, the FinRegistry and those sorts of things. But I had no idea. Probably I should read more, but there's so many hours in the day. We can do anything with a coherent, concerted effort. So you don't need a lot of data, but you need very accurate, validated data, and you need uncertainty quantification methods applied to the computational analyses that you do so that you understand the range of responses you get back. Because the thing about the virtual human is that it is using predictive modeling to make an actionable outcome. And in this instance, the actionable outcome is to inform the clinical decision-making. So we are there for at least 3 things that I can think of, but we need more. Thank you.
Thank you. Thank you very much. And with this, I would like to thank our speakers once more. I would like to thank you all for coming, and I'm closing this session. Thank you. Let's work for the brighter future.
Thank you, BAIBA, for having us.
Yes, thank you to our moderator.
Thank you. Thank you. Thank you.